Welcome to the Deep Dive. We are cutting straight through the noise today on a topic that causes immense daily stress for families navigating allergies. Soya.
Yeah.
It's a mandatory label, a common allergen, and yet it is also this global processing staple hiding in plain sight as refined oil and lecithin.
It creates a massive paradox, really. Soya allergy avoidance is challenging enough, but when you actually look at the clinical data, a huge amount of the resulting dietary restriction is just unnecessary.
Unnecessary? How so?
Well, it's often driven by outdated regulations or, confusing test results that don't reflect the real risk.
Okay, so that's the mission for this deep dive then. We're tackling three major areas where this clinical reality seems to clash with current policy or common practice.
Exactly.
First, we're investigating the true and maybe surprisingly low clinical risk link between peanut and soya allergy.
Right. Then second, we'll dive into the tolerance of highly refined ingredients like soya lecithin. This is a big one, especially for those with delayed non-IgE-mediated allergies.
Okay. And
finally, we'll look at the cutting edge, component-resolved diagnostics. This science is fundamentally changing how we test. It's like giving us a molecular magnifying glass to distinguish a true dangerous allergy from, harmless sensitization.
And the stakes here are incredibly high, aren't they? Soya is everywhere. So avoiding it often leads to severely restricted diets. Our sources highlight that for children with multiple food allergies, the median number of avoided foods on top of the main one was 3.
3 extra foods, imagine that.
Yeah, we're talking about removing most packaged snacks, desserts, things kids love. We have to keep that real-world impact in mind as we evaluate the data. Is this restriction truly necessary? Okay, let's unpack the first big clinical puzzle then, the peanut-soya connection. Allergists often see extensive IgE cross-reactivity in vitro. In the test tube, the antibodies look related.
Right.
So for you listening, the question is always, does that sensitization automatically translate to a life-threatening allergic reaction in a real person?
That is the crucial distinction, absolutely. And the research consistently shows the answer is overwhelmingly no.
No, really?
Yeah. While many children who have a clinical confirmed soya allergy also happen to have a peanut allergy, the reverse is actually rarely true.
Okay, give us the numbers.
So look at this. A recent UK study examined 68 children with challenge-proven peanut allergy. Proven.
Right.
Only 3% of those children had a concomitant confirmed soya allergy. Just 3%.
3%? Wow, that's tiny.
It is tiny. And that low clinical risk is consistent with, the existing literature. The confidence interval is quite tight there.
But here's where the conflict really hits then. Despite this really low clinical reality, the European Medicines Agency, the EMA, they currently advise that all medicines containing soya-derived excipients like refined oil or lecithin must be labeled as contraindicated for peanut-allergic individuals.
Correct.
Why this massive regulatory blanket over such a seemingly low clinical risk?
That's where we move from, let's say, clinically relevant risk to regulatory and, legal protection. This restrictive guidance stems from a specific point in history, a 2003 report from Sweden.
Okay.
It involved 4 deaths in peanut-allergic children, and these were linked, or thought to be linked, to high-level soya consumption.
So regulation is reacting to a small number of tragic fatalities.
Exactly. And while the EMA acted very cautiously, which is understandable, subsequent analysis suggested those fatalities might have actually been due to accidental peanut contamination, not the soya itself.
Oh, okay.
Generally speaking, soya is considered an uncommon trigger for fatal anaphylaxis. So the regulation, it's designed to prevent even the smallest possibility of harm, but the consequence is this overly cautious labeling that really complicates treatment and restricts diets, often unnecessarily.
So are we really willing to trade that dietary freedom and, ease of medical access for what might be a very small, perhaps even hypothetical, increased risk?
That's the debate.
If the clinical rate of co-allergy is only 3%, what does that tell healthcare providers to do in practice? What's the advice?
Well, based on the current evidence, experts generally do not recommend soya avoidance for peanut-allergic individuals unless they have a documented history of clinical reactivity to soya.
Okay, so only if they've actually reacted before.
Precisely. If a patient is avoiding soya merely based on sensitization tests or just general caution, the recommendation is often to pursue a supervised challenge.
To actually test it safely.
Yes. The data supports safely expanding their diet and improving quality of life rather than restricting it out of an abundance of caution that the clinical data simply doesn't support for most people. And this challenge of broad labeling brings us directly to soya lecithin. It's a key hidden ingredient, isn't it? Used as an emulsifier or stabilizer in just countless products.
Right.
For families trying to avoid soya, excluding lecithin significantly restricts the available choices, especially in, processed goods and baked items.
And the question of lecithin tolerance is especially critical for a slightly different patient population, right? Those with non-IgE-mediated allergy.
Okay.
For those focused on non-IgE issues, which are the delayed, often gastrointestinal symptoms like reflux, vomiting, or maybe malabsorption, not the immediate anaphylactic reactions, lecithin tolerance is a huge deal.
It is, because fears of triggering these delayed symptoms often necessitate total avoidance, just to be safe.
Yeah.
Which is why this GOMI study from 2019 is so fascinating. They specifically looked at children aged about 8 months to 5 years with confirmed non-IgE-mediated gastrointestinal soya allergy.
Tell us about their methodology. It was quite a rigorous design, wasn't it?
Oh, very. They ran a randomized, double-blind crossover trial. The children received 1.5 grams per day of soya lecithin baked into a biscuit on one day.
Okay.
And then a visually identical placebo biscuit on the other day. This double-blind, placebo-controlled design, it's really the gold standard for removing bias and confirming whether the ingredient truly causes the symptoms,
Absolutely.
Yeah.
And what were the real-world results? Did the lecithin actually trigger those delayed non-IgE gut issues?
The findings were incredibly reassuring. Out of the 20 children who completed the challenge, only one child that's 5% had a possible reaction to the active soya lecithin dose.
Okay, one possible reaction. What about the placebo?
Get this. The placebo biscuit triggered reported symptoms in 5 cases. That's 23%.
23% on the placebo versus 5% possible on the lecithin.
Exactly. It strongly suggests the symptoms, even when they occurred, were likely coincidental or, unrelated to the lecithin itself.
Right.
So the study found no statistical difference in reported symptoms between the active lecithin group and the placebo group.
So the conclusion?
The strong conclusion here is that for children with delayed non-IgE-mediated soya allergy, soya lecithin should generally be included in their diet.
Wow.
It allows for massive liberalization unless specific confirmed reactivity is proven through a proper challenge.
That's fantastic news for quality of life. Huge. But here we hit that regulatory dissonance again, don't we?
Mm-hmm.
Fully refined soya oil has been granted exemptions by the FDA and the European Food Safety Authority. They recognize the refining process removes the protein down to levels like 0.32 micrograms per milliliter. Basically nothing. Clinically irrelevant and safe.
Correct. It's considered non-allergenic.
So why does the EMA guidance on medicines containing soya derivatives, including lecithin, persist?
Oh, it comes down to that protein variance we touched on. While refined oil is consistently safe, the manufacturing process for soya lecithin is different, and the protein content can vary, sometimes quite widely, from maybe 28 up to 2,700 milligrams of protein per kilogram in some batches.
So it's the inconsistency.
Exactly. Even if 99% of lecithin is perfectly safe, regulators are focused on that potential 1% high-protein batch. It's a clear illustration of how clinical evidence on tolerance often battles with the regulators' need to account for all possible manufacturing inconsistencies, however rare.
Okay, so that conflict between broad labeling and individual clinical reality, it really brings us to the future of allergy testing. Component Resolved Diagnostics, or CRD.
Yeah.
This is the technology that helps us move past that crude sort of whole allergen testing and really distinguish true clinical allergy from mere sensitization, or that harmless cross-reactivity we started with.
And to use CRD effectively, you need to know the specific culprits, the individual proteins, the components. For soya, the primary sensitizers are often seed storage proteins, things like GLIAM5, which is a vicillin, GLIAM-M6, a legumin, and GLIAM8, a 2S albumin.
What makes one protein more important than another, diagnostically speaking? Like, why is GLAM8 considered such a critical marker?
GLAM8 is key because it's a storage protein, and crucially, it's highly resistant to heat and digestion.
Okay, so it survives cooking and processing.
Exactly. So if a patient reacts strongly to GLAM8, they are far more likely to react even to highly processed, cooked, or baked soya products. This makes GLAM8 a much more reliable predictor of a severe, true clinical allergy.
As opposed.
Well, contrast that with something like GLM4. That component is often responsible for the oral symptoms, itchy mouth, maybe some lip swelling experienced by people who also have birch pollen allergy. It's the classic pollen food syndrome.
Right, the cross reactivity again.
Precisely. So by measuring IgE antibodies to these specific components, you get a highly personalized risk profile, not just a yes-no answer.
Let's look at how researchers are trying to maximize this diagnostic power. There's a Japanese pediatric study by Maruyama from 2018. That's a great example.
Yes, a very neat study.
They took 91 children, used oral food challenges, the gold standard, to confirm who was actually symptomatic. That was 40 children, and who was just sensitized but asymptomatic, 51 kids. And then they tested 14 different soya components.
And initially, as you might expect, specific IgE levels to GLIOR8 showed the highest individual diagnostic value.
Because it's the heat-stable one.
Right. It had high specificity over 82%, meaning if you were positive for GLIOM8 IgE, you were very likely to be clinically allergic when challenged.
But they knew they could potentially do better than just looking at a single component. This is where the innovation comes in, isn't it? They decided to build a better diagnostic tool.
Exactly. A more precise one, they asked. How can we maximize accuracy while minimizing the confusing noise from some of those other cross-reactive proteins?
Like GLIM5, maybe?
Potentially, parts of it. So their breakthrough was developing a novel fusion protein. Instead of testing the whole GLIM5 protein, which can cause some confusing results, they cleverly took just a specific part, the N-terminal extension region of the GLIM5 alpha subunit.
Okay. A very specific
bit, and they chemically fused it to the high performer GLIM8.
So they created this hybrid molecule, the GLIM5A-AXTGAM8 fusion component. And what happened to the diagnostic accuracy when they tested that?
It shot up dramatically. The area under the curve, the AUC metric, which measures how reliably the test performs, where 1.0 is perfect.
Right, higher is better.
Higher is much better. It increased from 0.706 for GLAN8 alone, which was the best single component, up to 0.801 for the fusion protein. Even higher, 0.836 using relative values.
Wow, that's a significant jump.
It's a huge step forward. They achieved this boost by combining the highest specificity markers and essentially designing out the confusing cross-reactive parts. It shows fusion proteins are a really promising approach for molecular allergology.
So it allows clinicians to move beyond that generalized guesswork and make really precise evidence-based recommendations. Gives them confidence to liberalize the diet when those highly specific markers, like this fusion protein, are negative.
Exactly right. Hashtag tag outro.
Okay, so let's quickly recap the key actionable nuggets you can take away from this deep dive. First, the vast majority of peanut-allergic individuals do not react to soya in a clinically significant way. That co-allergy rate is really low, around 3%, which should push us toward individual assessment, maybe a supervised challenge rather than just broad avoidance based on peanut allergy alone.
Absolutely. Second, highly refined derivatives like Soya Lecithin appear safe for most children, particularly those with delayed non-IG-mediated allergies.
Right, the GOLMI study.
Yeah. The data strongly suggests that avoidance recommendations based solely on, say, sensitization tests or precautionary labels often lead to unnecessary dietary restriction, really without clinical benefit for many.
And finally, cutting-edge science is available now. If you or your clinician are using component-resolved diagnostics, CRD.
Which you should be asking about.
Yes. Pushing for testing specific to GLIM8 gives you the highest confidence score for identifying a true clinical reactor. And if that marker is negative, you can feel much better about liberalizing the diet.
And the concept of these fusion proteins combining markers like parts of GLIM5 and GLIM8, well, that represents the future of even more accurate diagnosis.
The research consistently highlights that the medical community has the data, really, to safely expand the diets of many allergic patients who are currently probably unnecessarily restricted.
It's about improving quality of life based on solid evidence.
Which leaves us with a provocative thought for you to explore. Given the verifiable power of specific component testing, like that GLIM5 5-EXT M8 fusion protein we just discussed, which provides diagnostic clarity right down at the molecular level. How quickly should broad, decades-old, population-level regulatory guidelines, like that EMA directive regarding medicines, be challenged and subsequently updated to truly reflect the most current clinical evidence?
It's that perpetual tension, isn't it, between broad protective policy and the push towards precision medicine based on individual risk.
Something to think about. Thanks for joining us on the Deep Dive.